So as I mentioned here, we talked about the importance of nucleotide, and they form the DNA, RNA, and the coenzymes, and the ATP, ADP. So this is the importance of the, okay. So the nucleotide structure, as I mentioned, are composed of nitrogen base, a pentose monosaccharide, and the phosphate. When you add the base and the pentose, it becomes nucleoside. When you add to the nucleoside, you add the phosphate, it becomes a nucleotide. You see at the end, zide, tide. Again, a nucleotide is composed of three elements: a base, what we call nitrogenous base because it has many nitrogens. A pentose is a sugar, and the sugar, it depends whether it's a DNA or RNA, and the phosphate. When you put a base in the sugar, you have a nucleoside. When you have to add a 1-phosphate, you have phosphate, you add it to the nucleoside, it becomes a nucleotide. So we will see what is the base, what is a pentose, and what is the phosphate. So as far as the base is concerned, you have two families of compound, purines and pyrimidines. You have two type of base, purines and pyrimidines. So the purines, if you look at this slides and this table, you have DNA and RNA purines, and here you have DNA and RNA pyrimidines. As you can see, both the DNA and RNA have what we call adenine and guanine. So adenine and guanine, these are purines, and they are both found in DNA and RNA. And if you look at the pyrimidines, both DNA and RNA have thymine and tyrosine, but the difference is in the RNA, it has the uracil, and the DNA have a thymine. I will repeat. The purines, the DNA and RNA, both have the same purines, adenine and guanine. However, the difference between the DNA and RNA is the uracil. Uracil is only found in RNA, not DNA. So this is the only difference between the DNA and RNA as far as the base is concerned. Again, if you want to see whether this is a DNA or RNA, you look at the bases. If you find uracil, is RNA. If you find thymine, it means DNA. Thymine is only found in DNA, and uracil is only found in RNA. This is not to memorize. In fact, don't worry about the structures. You don't need to memorize them. The only thing you should know, what are the two families of bases, which is purine and pyrimidines, and what are they, and how can we differentiate between bases found in DNA or RNA. As I mentioned, it's very easy. Look at the uracil. If it's there, is RNA, and if a thymine is a DNA. I will give you a tip how to remember these bases. Basically, if you look at the adenine and guanine, it's adenine start with A and guanine with J, and these are purines. You say pure as gold. Purine, remember purine, pure as gold. And then you remember pu is purine, as is adenine, as gold, guanine. And if you know these are purines, the rest are pyrimidines. These are the way we number the bases. You don't need to memorize it at all. It's just to have an idea how we number, because later on you will find 1, 2, 5. So you wonder what is the numbering, and this is how they number. Later on, when you read, if you want really to be more in details and you want to know where is the number 6 or 5, carbon 5, you can come back in the slides. But this is to ignore pyrimidines, cytosine, uracil, thymine, etc. So this is the numbering. As we mentioned here, we said there is a purines, pyrimidines, and we have minor purines. Why we call them minors? These are not involved in the formation or synthesis of DNA and RNA. That's why they call them minor, but they are important in other aspects that we will see later on. So you have purines, pyrimidines, which are the adenine, guanine, thymine, cytosine, and uracil. These are the major, if you want to call them bases, nitrogenous. You have a minor purines with are hypoxanthine, axanthin, and uric acid. These are product of metabolism of adenine. So basically adenine and guanine. So they do not enter in the formation of nucleic acids. So basically when you degrade adenine and guanine, the end product of them is you either get hypoxanthine, axanthin, and uric acid. So if they ask you a question, what is the product of metabolism of adenine or guanine, your answer will be hypoxanthine, axanthin, and uric acid. And they are coming from the adenine and guanine, not from thymine, cytosine, or uracil. Now, we looked at the base, which is purine and pyrimidines and the minor bases. Now we look at the pentols. If you remember, we talked about this, base, purine, pyrimidines, minor. We are going to talk about the pentols now, and then the phosphate. So the pentols, what type of pentols do you have? This is a chemistry recall. It's a ribose. So the type of pentols, penta, it means 5. It's a ribose. So what are the two types of ribose? We have the two types of pentols. We have ribose and the deoxyribose. So this is your ribose, and in the RNA, and in the DNA, it is the deoxyribose. I repeat. So the pentose sugar, it's a ribose. We have two types of ribose. The ribose itself in RNA, and as you can see here in the 2', there is OH, hydroxyl. However, in DNA, in 2', you have only hydrogen. You have lost your oxygen. That's why we call it deoxy. Deoxy, it means without oxygen. Why 2'? It means at position 2. Now, you might ask me why you are doing a prime, this prime. We do the primes in order not to get confused with the base, because the sugar will be attached to a base. And if we don't do the prime, then maybe I will show a lecture. When I found the picture, I will tell you. You see, this is for instance a sugar, and is attached to this base. So if you don't write 1' here, how do you differentiate between the 1' here and the 1 in here? If I write 1, 1, do you get my point? We do the 1' to differentiate. The 1' is in the sugar molecule, and the 1 without a prime is in the base. So we've seen now the pentols. You have two, you have the ribose in the RNA and deoxyribose in the DNA. If they ask you which of the following belongs to the RNA, ribose or deoxy, you will say this is the ribose. And you need to know that the ribose has OH at 2' and deoxy has only H at 2'. Now, when I started the lecture, I mentioned nucleoside. I mentioned the nucleoside, and we said the nucleoside is when you have a base plus pentose only, without phosphate. So a nucleoside is the addition of a pentose sugar to a base produce a nucleoside. So if the sugar is ribose, you call it a ribonucleoside. And if the sugar is deoxyribose, you call it deoxyribonucleoside. So if I tell you what is ribonucleoside, you say this is a sugar plus a base. And it is in the RNA because it's a ribo. And the deoxyribonucleoside, this is in the DNA because it has deoxyribonucleoside sugar, deoxy. So this is the ribose, again with the OH, and this is deoxy without the O. If you add a base, this is the cytidine, called the cytidine. This is a base plus cytidine, and this is the deoxyadenosine, adenine. It means this is the adenine, and when you add a sugar from the deoxy, it becomes deoxyadenosine. I know it's a bit confusing. You see here, atidine, it should be inosine. As I mentioned here, a base plus a sugar, it should be zyme. But in this case, it's just difficult to call it cytosine, cytosine. So this is different from the ozine. It's more suitable and easy to spell out, which is cytidine. So be careful about this. In general, the rule is you add zide, but sometimes it doesn't apply, and then it's cytidine. So this is in general a nucleoside, which is a base plus a sugar. And in the RNA, you find the ribonucleoside. In DNA, you find deoxyribonucleoside. For instance, if this is A for adenine, J for guanine, C for cytosine, and U for uracil. So if you want to add a sugar to this base in the RNA, it becomes adenosine. You see, zine. If you want to add a sugar to guanine, it becomes guanosine, but it doesn't apply to, as I mentioned, uracil and uridine. So these are uracil, it becomes cytidine, it becomes uridine, and cytosine, it becomes cytidine. That's why when I started to tell you, it's very important to start and focus on the nomenclature of the bases and nucleoside and nucleotide, because later on I will use guanosine. You should know when I say guanosine, it means guanine plus sugar. I take you back again here because it's very important. Guanine plus sugar, it becomes guanosine. Adenine plus a sugar, it becomes adenosine. Thymine, thymidine. You see here is different. Cytosine, because it has already zine, it becomes cytidine. And uracil, it becomes uridine. Okay, so of course, you will have a lot of information, but that's why when you go home and sit down and look at it carefully, and it will be easy to start picking up. Now, this is as far as the RNA. And as we mentioned, if we want to name the DNA, we have to add the deoxyribonucleoside, because we are now talking about DNA. We don't say ribonucleoside, we say deoxyribonucleoside. So if you have an adenine and you want to tell me this is from DNA, you have to add a deoxy. It becomes deanosine, deoxyadenosine. Deoxy, it means the sugar is deoxyribose, and adenosine, it means adenine plus your base plus your sugar. So this is the base, the adenine. When you add to it the ribose, it becomes deoxyadenosine, and etc. The compound deoxythymidine, usually is called thymidine without a D. This is just for your information, because the RNA hasn't got the thymidine, because the prefix deoxy is often left out since there are no precursor of thymine nucleoside involved in RNA synthesis. Because there is no thymine in the RNA, so it's clear when you say thymidine, we understand as this is for DNA, not for RNA. So this slide is about how to name ribonucleosides, which are nucleoside. Nucleoside is a general word. Nucleoside, it can be ribonucleoside for RNA, or it can be deoxyribonucleoside for RNA. And it depends on the base, you name your nucleoside. This is a table as an example to make it easy for you to understand. Examples, if you want to practice, adenine plus ribose, it becomes adenosine. Guanine plus ribose, it becomes guanosine. All these are nucleoside. Uracil plus ribose, uridine. You see here is a bit different from zine. So it becomes dine. Cytosine plus ribose. There is an exception here. If you look at the minor bases, hypoxanthine plus ribose, here is something completely different called inosine and inosine. So please remember this. Later on, when you see inosine, it means hypoxanthine plus ribose. This is a minor base. Xanthine plus ribose, this is guanthosine. So here is clear, but the difference, the only exception here. Now this is a ribonucleoside. It means we are talking about RNA. When it comes naming the DNA, we use deoxyribonucleoside. It's the same principle. Adenine plus deoxyribose is deoxyadenosine, etc. I put another slide to make it even clearer. You see the base is adenine, nucleoside is adenosine, and if we want the nucleotide, we call it adenosine, let's say monophosphate. There is only monophosphate. You see, adenosine is we are here. When I add the phosphate to this, it becomes adenosine monophosphate. Guanosine monophosphate. And this is a nucleotide. A nucleotide is different from a nucleoside because it has a phosphate in it, etc. Inosine monophosphate is the same principle. This is about nucleotide. This is a nucleotide. Nucleotide about you have your base, you have your sugar. These two are nucleoside. When you add a phosphate, it becomes ribonucleoside monophosphate. You see here, the nucleotide monophosphate, and for nucleotide, if you add another 2-phosphate, it becomes ribonucleoside diphosphate. If you put 3, it becomes ribonucleoside triphosphate. So the addition of one or more phosphate groups to a nucleoside produces a nucleotide. So the first group here is what we call an ester linkage. This is when there is the oxygen from the sugar molecule linked to the phosphate of the bond to the phosphate, it becomes, we call it ester linkage. So this is what I've just mentioned when you add 1 or 2. This is just for your, if you want to see the linkage, ester linkage clearly, this is the ester linkage. Now, it's like we finished with the introduction of the how to name nucleotide, nucleoside and nucleotide, what type of a bases we have, what type of sugar we have, and what is a nucleotide. Now we want to look at how we are going to synthesize purine nucleotide. Again, I will remind you, purine nucleotide, it means adenine monophosphate, guanosine monophosphate, because we are talking about purines. So the purines bases are adenine or guanine. So this is very important in order to be able to follow. Purine, they are pure as gold, adenine and guanine. They are nucleotide, it means we are talking adenosine monophosphate or diphosphate or triphosphate or whatever. So this is what we want to know, how we synthesize this nucleotide. First, we want to see how the purine ring itself is synthesized. How can we synthesize the purine ring? Why is called ring? Because it has a double ring, as you can see. This is all why they are called the ring. This is one ring of 5, and this is a ring of 6. So the N atoms, these nitrogen atoms, they come from aspartic acid, glycine, and glutamine. This is the importance of having amino acids in your diet. You see how important the amino acids, they are involved in the formation of bases that makes our DNA or RNA. So this question has been asked before, so please make a note for these slides. The N atoms, or maybe the purine ring of nucleotides, purine nucleotides, the N atoms come from aspartic acid, glycine, and glutamine. The carbon comes from carbon dioxide, and the N10-formyl tetrahydrofolate. So as you can see here, the carbons comes from the CO2 and the formyl tetrahydrofolate. So you might have a question asking, what is the main component of a purine ring? Which of the statement is correct? The purine ring is synthesized from amino acids, glycine, aspartate, and you put something else, and then you should be able to know that it's only aspartic acid, glycine, and glutamine, that they are important in the formation of a purine nucleotide. Now, what is a de novo synthesis? De novo, it means from scratch. It means if you want to build something from scratch. There is de novo, it's a French word, de nouveau. And there is a salvage synthesis. Salvage, the English means rescue. So rescue, it means you make nucleotide already made. There are pieces and just you are going to gather them together. However, de novo, it means you are starting from the beginning. It's like, let's say you want to build a car. You need to start from the beginning, make the engine, make the wheels, everything should be from the basic. This is de novo. However, if you want to make it from to salvage, for instance, you have already the engine is already there, the wheels are already there. You just come and build up together. So this is salvage. Now you will have first step is to make what we call PRPP, the 5-phosphoribosyl 1 pyrophosphate, and this you should know this before because this is a molecule that you've seen in the pentose phosphate pathway in the when you did the carbohydrate metabolism. So this is already done. If you want to recall, you go to your lecture and you find out how to get the PRPP. So this is, you get it from the pentose phosphate pathway. The purine, if they ask you where is the de novo synthesis of the purine, the site is in the liver, is the major site of purine synthesis. This is very important point. So this is the PRPP. How it's synthesized, again, this already been done in your before. So if you have a ribose 5-phosphate, and then what you do, you take 2-phosphate from the ATP plus magnesium, and you put it in the ribose 5-phosphate. You see, this is number 5. That's why they call it ribose. This is a sugar fibose. 5, this is the position 5, phosphate. When you put these two phosphate, when you have a 2-phosphate, you call it pyrophosphate. Pyro means 2-phosphate. So you take 2-phosphate from your ATP, you put it into your ribose 5-phosphate, and you will have 5 phosphoribosyl. This is a phosphoribosyl. When you have zyl here, it means a radical. So you have 5 phosphoribosyl, 1, this is position 1, pyrophosphate, it means 2 phosphates. So I will take again 2-phosphate from my ATP with the magnesium in the reaction. The enzyme responsible is PRPP synthetase. This enzyme will take 2-phosphate from here, from the ATP. We put it in the ribose 5-phosphate at the position number 1, and I will end up by having 5-phosphoribosyl and 2-phosphate called pyrophosphate. So what activates this enzyme is the inorganic phosphate, and what inhibits this enzyme, these are what we call the end product of the purine synthesis, which are adenosine monophosphate, guanosine monophosphate, and very important compound, ionosine monophosphate. Now, and it's very clear, because if you have a lot of nucleotide, then you don't want to synthesis, you need to inhibit. If you have less AMP, GMP, inosine monophosphate, the PI will activate this enzyme, which make more AMP, GMP, and ionosine. This step is not involved in the purine synthesis, but rather is a step before the real real synthesis. But you need to know this is the beginning. This is the first step. We can consider this is the first step of purine synthesis. Okay, so this is considering the first step of purine synthesis, the synthesis of 5' phosphoribosylamine. It's very easy. If you look carefully how it's formed, it's easy. This is your 5, this is position 5, phosphoribosyl. As I mentioned, when you see zyl, it's a radical. This is phosphoribosyl. Don't worry if the alpha is the same as 5. You will 5 alpha, and here is beta. Alpha is number 1 at the position 1, and beta at the position 5. So these compounds, see the 5 prime, this is 5, phosphoribosyl alpha, pyrophosphate, this is the alpha, pyrophosphate, or number 1. What you do, you take this 2-phosphate, the pyrophosphate, and then an enzyme comes called the glutamine phosphoribosyl pyrophosphate aminotransferase. The aminotransferase, it means is going to transfer amines from the amino acid glutamine. It takes 2 amines and put them at beta position here. See, this is the amine, the 1 amine, sorry, not 2. The amine, put it into the beta position, and the glutamine will become glutamate. And the end product is the 5' phosphoribosylamine or beta amine. You can ignore the beta. You see, synthesis of 5 or beta. They are both nomination, nomenclature is the same. Now, you might ask me, you tell me, I told you what you need to know on the previous slides. You tell me, oh, doctor, what can we do about this? I tell you, what you need to know is the PRPP, you see here the glutamine and PRPP are important to make the phosphoribosyl beta-amine. So this is important. If they ask you how to make it, you say from PRPP and glutamine. This is one. What is the enzyme involved? So this is the enzyme. What is most important is this step is the committed step in the purine nucleotide biosynthesis. Now, I hope you have already know what is a committed step, because this should be taught at the beginning of the course. Does anyone know what is committed step? Okay, the committed step is the step that dictate how the speed of the reaction. It means if this reaction finish, it means the whole reaction will follow. You cannot go back. And this is increases, everything will be increased. And if this stops, everything will stop. That's why it's the committed step. Committed, it means once it starts, that's it. Everything should follow. That's why this reaction is very important. So the rate of the reaction is controlled by the intracellular concentration of PRPP. So let's continue. So this is the second de novo synthesis, as I mentioned. This is one of the major important reaction. Now I'm going to send you, let's look at the PRPP. How the rate of reaction is controlled is it depends on PRPP. As I mentioned at the beginning, there is de novo synthesis and salvage synthesis. If the PRPP is too high, you start from scratch synthesis. If the PRPP is low, you start from de novo. So this is, you have to salvage. I repeat, if the PRPP is high, you start de novo synthesis. If the PRPP is low, you have to salvage. So if they ask you when the de novo synthesis starts, you say when the PRPP is high, and they tell you if the PRPP is low, which one the reaction will follow, you say is the salvage. And is the same as the first reaction. This reaction is inhibited by the end product, which are AMP, GMP. You see here now I'm saying AMP and GMP. If you don't grasp the name, that's it. You are going to ask yourself, what is AMP and GMP? So, and ionosine monophosphate, which are the end product of the pathway. What it means, the end product, I'm going to send you this. So don't get scared. You don't need anything to worry about this. This is just to tell you how complex the purine synthesis is concerned. What we are looking for in our case is just, let me show you the, we are looking at just this step, this one. So this is step we are looking at. Can you imagine these all are the steps to follow? And the end product is the ionosine 5' monophosphate. So this is the end product. And after this, after the AMP will give AMP and GMP. That's why we call them end product. The end product of the purine. We start from here, and at the end we end this reaction. So this is what we call this is what I've been talking early on about the main committed step. So if this starts, all reaction will follow, all these reactions. So we are looking only on one step. There are 9, I think. So we are not interested. We're not going to look at this. We looked at this, and the one previously, as I mentioned, you have already looked at in the pentose phosphate. So you need to know about this reaction, the details with the first one, and you need to know the end product, which are AMP, GMP, and AMP. Now, there are some, why these steps are important. We have some medication here we use, and the way these medications are important because they interfere with some enzyme within this reaction. For instance, what we call sulfonamide, when you will do the microbiology, you will see this antibiotic, and it interacts with the tetrahydrofolate. It inhibits the tetrahydrofolate, and therefore the bacteria cannot grow and die. There is another medication called methotrexate. It has the same action, but the different enzyme, so which limits the amount of tetrahydrofolate available, the same as the sulfonamide, and then will inhibit the mammalian. Mammalian, it means the human cells, and therefore is used in chemotherapy. So the importance of knowing this reaction, it helps people or scientists to develop drugs, try to treat some diseases. In this case, as I mentioned, you have an antibiotic used to kill the bacteria, sulfonamide, and you have a methotrexate, another compound, but used in cancer. And both of them inhibit the tetrahydrofolate. So don't worry about this. Now, one of the questions that has been asked before, what is the parental or the parent of purine nucleotide? The answer is the AMP. Can you see the so the ionosine monophosphate, this compound here, this one, so this is the parental or parent of purine synthesis. So be aware about this point. It's very important in the future. So the ionosine monophosphate is the parent of purine nucleotide. And as I mentioned, there are 9 steps. We are not interested in these steps. What is important for us after the 9 steps, after all these 9 steps, we end up of ionosine monophosphate, which is important for us. So there are, as I mentioned early on, there are some what we call inhibitors of purine synthesis are used to treat bacterial infection or cancer. And I told you there is what we call sulfonamide, and it inhibits the dihydropteroate synthase, and some folic acid analog like methotrexate and trimethoprim, and both, one of them as well, trimethoprim, is an antibacterial activity, and the other is anti-cancer. So basically, this is, if you want the details, this is how the sulfonamide will act on to kill the bacteria, and trimethoprim and methotrexate inhibit the dihydrofolate reductase to inhibit the DNA synthesis. So as far as the medication is concerned, you have to be aware about 2 things. The sulfonamide inhibits the dihydroteroate synthetase, and therefore the DNA, the bacteria cannot multiply, and therefore they cannot synthesize DNA, and therefore it dies. The same as the trimethoprim and methotrexate, they inhibit the dihydrofolate reductase, and therefore there is no tetrahydrofolate synthesis, and there is no purine synthesis. And if you don't have the purine synthesis, it means you cannot make DNA. This is the
Doctor.
No. Yes.
Is the enzyme for memorize or not?
Yes, that's anything. I'm skipping many things. So anything I mentioned is important to know. things that I don't have mentioned, as I mentioned before, don't look at them. What biochemistry is about most important enzymes and committed steps. So that's why there are 9 reactions. I didn't talked about them at all. I told you about only 2 reactions out of basically 10 reactions. So I've selected only 2 reactions out of 10 reactions. So if you can you imagine if I told you 11 reaction with 11 enzymes, so you have to deal only with few enzymes here. So this is we said they start by ribose 5 phosphate. We said we make the PRPP. This is the first reaction I mentioned at the beginning for you. So this is the first reaction I told you, this one. Then the second reaction I told you again. You see, only you have 2 reactions. If you want to say what do you memorize, this is your first reaction and the enzyme, and the second reaction and enzyme, and we didn't talk about all this. So you need to know the enzymes, what inhibit them, and what trigger the synthesis. So what is the positive or negative? So once this is the end product, IMP, inosine monophosphate, okay? From the inosine monophosphate, you get your AMP or you get your GMP, which are the purine nucleotide. So basically, we started from a base, from the RPPR, RPRP, PRPP. We start from this, and we end up at the IMP, and from IMP, we get AMP and GMP. This is very, very simplistic slides that helps you to know what to memorize. Just this is a feedback control. Once you have a lot of AMP, of course, this will inhibit this enzyme to stop the production. It's the same, you have a lot of AMP or GMP, will go back and inhibit these enzymes to stop the synthesis. So this is what you need to know about this is conversion of AMP to AMP. This is just, as I mentioned again, you need the end product. You have the AMP. This is your AMP. You see how I'm not talking about many things here. See what I'm telling you. This is your AMP. You get from it AMP and GMP. That's it. I'm not telling you this reaction, or these, the enzyme, nothing. I want you to know that you get AMP from IMP. You get GMP from AMP. So this is all you need to know. So this slides is the same as this. Just focus on what I've just said to you. From where you get your AMP, adenosine monophosphate, is from your AMP. From where you get GMP is from your AMP. Okay? Here, what you need to know, the first reaction, second, their enzyme, how they are inhibited. And as you can see, this part here, this part is the same as this one. This slice. So now if you have an AMP monophosphate or GMP, how can you get to the diphosphate? Adenosine monophosphate, when you add to it a phosphate from the ATP, it becomes adenosine di. Mono, it becomes di. And when you take out one phosphate from here, it becomes di. So one di, it becomes 2 adenosine diphosphate. Is here GMP, if you take one phosphate from here and give it to GMP, it becomes GDP, di. And here, it's left with adenine, di, because you are given one phosphate here. You see here, there are two different enzymes, adenylate kinase, and you know in the previous lectures that kinase, it means phosphate, given phosphate. Adenylate because it's adenine, guanylate because it's guanine. Now, how do you go from diphosphate to triphosphate? Is the same. You take the one phosphate from ATP, you give it to GTP, it becomes guanosine triphosphate, and this is left with adenosine diphosphate. The difference between these two enzymes, these and this, here they are, the enzymes are specific to the base, adenate to AMP, to adenine, guanylate to guanine. However, in the nucleoside diphosphate kinases, they are not base specific. They have a broad spectrum. It means this enzyme can work on the guanosine and adenine both. But the monophosphate kinase, they are specific to their bases. So this explanation is all what is in this slide. What is time so far, Osama?
I think 20. 20
We
minutes.
did 20 minutes, okay. So I want to take 2 minutes or maybe 5 minutes to summarize, to tell you what we've done so far, because I know it's a lot of information and what we call a rough subject. Some students come and they think the lecture is not well explained or whatever. In fact, they don't realize that there are different types. There are lectures that are easy. You talk about anatomy, you have 5 fingers, you have the muscles, you memorize, and it's easy. Biochemistry is a rough subject, but by nature. So this is why many students, they don't like the biochemistry, because it feels they are difficult to understand. But this is the nature of the lecture itself. So you need to know this. So international student, even when I taught in Britain, they found the same difficulty with students usually they don't like memorize enzymes and etc. But we try to give them few reactions, 2 out of 10, 3 out of 10 reactions. We highlight some aspect and that's it. So this is how we can help you to shortcut all of these. But things that we can't do is, this is the nature, طبيعة الدرس. It's not like physiology, anatomy, or whatever, or Arabic, or this is. So the deoxyribonucleotide, how do you make them? And again, you see what if you look carefully about the way I'm giving you the information, if you take notes, it will be easy for you. The first thing you should know, we talked about ribonucleotide found in RNA. How do we make the deoxy? How do you make those for DNA? First, what you need to know, what is the enzyme responsible for converting ribonucleotide to deoxy? It's called the ribonucleoside diphosphate. So, or also called ribonucleotide reductase. So, in your question, might ask which of the following enzymes required to make your deoxyribonucleotide, and we give you many enzymes. The only thing you should know there is an enzyme called the ribonucleotide reductase that makes the deoxyribonucleotide. So this is the only information on this. Now, this enzyme is composed of 2 non-identical subunits, R1, R2. This is all you need to know. It has 2 subunits. Now, you see here, if you look adenosine diphosphate, this is for RNA. We want to convert it to adenine deoxyadenine phosphate for the DNA. So we need to call this enzyme to make the job. In the RNA, we have guanosine diphosphate, and we want to make deoxy for DNA. We call this enzyme again. So this enzyme, ribonucleotide reductase, is responsible for making the deoxy from the ribo. So it makes the deoxyribo from the ribo. So this is the other point. This is an explanation of ribonucleotide, to how they are converted into deoxyribonucleotide. As we mentioned early on, when we looked at the purine synthesis, we said that at the end of the 10 reactions or so, we end up with the inosine monophosphate, which is the parental molecule. And we also said from the IMP, we get AMP, GMP. So all these are purine nucleotide, and they are the end product of de novo purine synthesis. If you look at, once you have the IMP, you get the AMP. This line, can you see, is for the RNA. The GMP, this line is for the RNA. So once you make your ADP for RNA, if you want to make your DADP, you call for the enzyme 1, which is ribonucleoside diphosphate reductase. Then from deoxyadenosine diphosphate, you get deoxyadenosine triphosphate, and then you make your DNA. The same for the guanosine monophosphate, you make your guanosine diphosphate, then triphosphate, then you make your RNA. If you want to make your DNA, once you are in GDP, you use the enzyme ribonucleoside to make your DNA. So basically, this is again, a reminder. We started at the beginning, if you remember, if you recall, at the beginning, we start with ribose 5-phosphate that you already know it comes from the pentose phosphate pathway. We said there is an enzyme called PRPP that helps you to make the PRPP. Then the PRPP, we end up by the 5' phosphoribozolamine. Then we went through a 9-step reaction to reach this AMP, and from this AMP gives you the AMP and GMP, and from this monophosphate you get diphosphate, and as you know, you need triphosphate. The DNA or RNA are made up of triphosphate. So I think this slides explain the whole thing, that if you want to prepare for your exam, it's clear, you know, I need to know this reaction. I need to know this reaction. I need to know the end product. I need to know from this I get this, and how to get from RNA to DNA, this enzyme. So, and it's very simple. From these two slides, these slides can help you to summarize the whole slides we've seen before. Again, it's the same here. How do you make the TTP? Because if you see here, the DNA, we said the DNA has a thymine monophosphate. See the nucleotide triphosphate, the thymine. Let's take it. If you look at here, we have this is RNA. Adenine give you deoxy. Guanine give you deoxy. Guanosine. Cytosine give you D. And uracil give you DUTP. Someone might ask, oh, doctor, the DNA needs DDP, and it's not here. The answer to this is in this slide. This is from where we get our DDP, is from UDP of the RNA will be converted to DUDP, then DUMP, and comes to the DTP. So what is important in this? What is the only information you should know is the DTP that makes the DNA comes from the UDP. This is the most important information from this slide. UDP give you the UDP, UDP, give you the UDP from where you get the DTP. I repeat, if they ask you from where you got your DTP of the DNA, the answer is the UDP, and that's all. Which enzyme again is used is the ribonucleoside. You see, you have only two information to know from the slides. Now, we've seen how purines have been synthesized. Now, how the nucleotides are degraded. So what is the first information? What we eat nucleic acids. I mean, in the food or meat or whatever, there are some DNA, RNA. When you eat it, in the small intestine, it will be degraded. Where the nucleic acids are degraded in the small intestine, then there are some enzymes from the pancreas. It will degrade, hydrolyze the nucleotide to nucleoside and then the base. You see, we know that the base will make nucleoside with the sugar, and when you add the phosphate, you get a nucleotide. So when we eat a nucleotide, we go to the nucleoside and then go to the base, and then from the base, the cell will absorb it. Okay? So the base itself, when you have the base, this base will be further degraded. The base, we mean purines, adenine, and guanine, will be further degraded to give you the uric acid. So if they ask you, what is uric acid? Your answer is the uric acid is the end product of purine metabolism or purine degradation, more accurately. I repeat, what is uric acid? Uric acid comes from the degradation of purines, which are adenine and guanine. Again, this is just for to know how DNA is degraded. You eat DNA, RNA. In the stomach, the low pH will denature the DNA and RNA. Then the enzymes, pancreatic enzyme will make it into oligonucleoside, then to mononucleosides, then to nucleosides, and then to the basic pyrimidine or purines. So it means adenine or guanine, or cytidine, thymine, and uracil, and the purines will be degraded into uric acid, and the pyrimidines, some of them will be going into the circulation again. So what information is important here? You need to know that the DNA and RNA received from food will be degraded into pyrimidines and purine bases, which will be absorbed. So the dietary purines are generally converted to uric acid by intestinal mucosal cell. So how how uric acid is formed? So basically, all these steps leads to uric acid formation. Are we going to look at all these steps? No. What you need to know is just this step, the last one, that once you have a end product which are IMP, ionosine, and the AMP, IMP, and GMP. These are our purines. Okay? All these will be end up as xanthine. You need to know what is the end up of the IMP, AMP, GMP will end up xanthine. Xanthine will be converted to uric acid. Why is important this reaction? Because there is a disease called gout, nawras, I think, called in Arabic, or naqras. So and it's very common. So it's important to know that this disease comes from, let's say, hyperuricemia, uric acid increase, uric acid in the blood, because there is a there is a lack of secretion of uric acid. So the kidneys do not secrete properly the uric acid, and therefore is going to be built up in the body and leads to a gout. So we look at the gout. So it's a disorder characterized by high level uric acid. So if I ask you a question, what is a gout? You tell me gout is a increased level of uric acid, which is the end product of catabolism of purines, adenine and guanine. So you have a high, what we call uric acid in the blood, called hyperuricemia. This hyperuricemia will lead to monosodium urate crystals build up in the joint, and therefore it will have an inflammation response. So basically, people, they will have like nodules in their fingers, or particularly in their toe, big toe. So you will have a formation of nodules in the fingers or in the big toe. As far as people that have gout, they usually have kidney stones. So gout is an increased level of uric acid due to a lack of secretion from the kidney. If there is uric acid, high secretion, a high buildup of uric acid in the blood, then there is a increase of deposition of monosodium urate crystals in the joint, and therefore the inflammation, plus stones in the kidney. How to make the diagnosis of the gout? We do the aspiration. Aspiration, basically, we take a needle and you put it in the joint and to take the liquid, and then we look at it under the microscope and we will find these sodium crystals. Now we have finished with purines, synthesis and degradation. The pyrimidines, they are not as important as the purines. So basically there is no much on it, but what we need to know, again, is the same as the purines. You need to know how the purine ring is formed. So basically these are the component of purine synthesis, are again from amino acids, but the amino acids some of them are different from those in the purines, and they have the same, again, the nitrogen, the carbon comes from the formal tetrahydrofolate. So very important. I have asked question before in these slides. I might tell you what's the purines ring, from where the carbon dioxide comes, or what are the amino acids, what's the difference between the amino acids found in purines compared to pyrimidids. So please look at the slides and compare it with the one of the purines. There is one important enzyme in the whole reaction. This is the reaction of the purines, which are not going to go through them all. I'm just putting the slide to have an idea, but there is definitely one enzyme you need to know about it, and because it's important in the committed step reaction, and because you have already one type of this enzyme. This enzyme is called carbamoyl phosphate, and you already know, and you have already seen in your previous lectures, there is one enzyme called carbamyl phosphate, but in this is found in the mitochondria, and you did it when you looked at the urea cycle. However, the carbamyl phosphate we are looking at today is called number 2, not number 1. So I might ask you a question, where do you find carbamyl 1 or 2? Or I will tell you the source of nitrogen from the carbamyl 1 or 2. So this slide is important as far as this enzyme is concerned. So what is the most important slides in in the pyrimidine reaction is this one, the first one, and this is where the carbamyl phosphate synthase 2 is involved. So you need to know what inhibit and what. So
Doctor,
now
we have just 1 minute. Can we complete next week?
I will finish it. I will finish. So basically, this is the most important and step in this reaction. Next lectures, I will be more quick than this. So, so basically, this is the most important reaction you need to know. And from where you get your UTP, your CCTP, cytosine triphosphate, you will get it from the UTP synthesis. So if they ask you from where you get your CTP, basically you get it from UTP. And this is again, how you synthesize thymidine monophosphate. This is the reaction from the synthesis of thymidine monophosphate. You get it from the UMP. You get DNTP. So basically, I expect from you to know from where, when I ask you from where you get your DNTP, you say it's from the DNP and the enzyme that they are involved. And there are some, of course, some diseases related to this. It's the same explanation. Okay. So basically, I'm done with this and I will see you inshallah next week.